this lesson, we are going to be looking at the light-independent reaction. So this is part 3 of our unit on photosynthesis. Just as a matter of review, I just kind of want to touch on where we've been. So if we look at photosynthesis, we looked at the light-dependent reaction, or sometimes called the light reactions. And in the light reaction, we're going to take the water, we're going to split it, and we're going to use the hydrogens from the water to generate a whole bunch of energy in the form of ATP over here and NADPH. From that, we have a waste product created called oxygen. And here's our waste product, which is kind of a convenient waste product because then we'll inhale that in cellular respiration. But where we really left off is this idea. So the whole purpose of the light reaction was to generate energy. The light reaction is a generator. It generates ATP energy and NADPH energy. And that energy goes in to power this Kelvin cycle, or also known as the light-independent reaction. So in the Kelvin cycle, we're going to take that energy that we created, and we're going to form carbon dioxide into sugar. Okay? So we're going to power the Kelvin cycle, which turns carbon dioxide into sugar. Now, in this, we're going to take carbon dioxide and we're going to convert it to glucose. But before we convert it to glucose, we're going to turn it into a molecule called G, and I'll write it down here, G3P, and that's a precursor to glucose. It's 1/2 of a glucose molecule. And we're going to do this through a cyclical process, which means the thing that we end with is the same thing we start with, which is great because all we need is our inputs again, and then we can redo it. We don't have to start all over and rebuild everything. We can just input more carbon dioxide and more energy and run the process again. So it's a cyclical process. That's why we call it the Kelvin cycle. I know this graphic down here is hard to see, and we'll zoom in on it in just a little bit, and we'll talk about what's going on here. So what's eventually going to happen? So the short form of this is this. We're going to take 3 carbon dioxide molecules, and we're going to convert that into 1/2 of a sugar molecule. Because we know that sugar is C6, right? So sugar has 6 carbons, 12 hydrogens, and 6 oxygens. So we know that we have 6 carbons, right? Well, if we take 3 carbon dioxide molecules, if I take 3 molecules of carbon dioxide, CO2, I have 3 carbons. 3 carbons, so that equals 1/2 of a glucose molecule. So 3 carbon dioxides, essentially, is a good part of 1/2 of a sugar molecule. So that's what we're going to do. We're going to take 3 carbon dioxides and build 1/2 of a sugar molecule, and we're going to use energy in the form of ATP and NADPH to do that. Now here is our process, and I'm just going to talk through it. Here's sort of the first step right here. So if we look here, we have this molecule called ribulose 5-phosphate. And they say 5 because it has 5 carbons. 1, 2, 3, 4, 5. And it's going to bond with carbon dioxide over here. Right here's, they're going to add phosphate to it to make ribulose 1,5-biphosphate, which has 5 carbons. 1, 2, 3, 4, 5. Okay, and it's this RUBP, they call it, ribulose biphosphate, RUBP. They are going to attach carbon dioxide. So look here, here's our carbon dioxide molecule. It's going to get attached to RUBP using an enzyme called Rubisco. Rubisco is the enzyme that attaches carbon dioxide onto RUBP. And it creates this molecule here, which is going to be a 6-carbon molecule. Okay? This 6-carbon molecule actually breaks apart into 2 3-carbon molecules. And then we add some, so phase 1, so if we talk about these phases here, phase 1 is carbon fixation. This is where we're going to attach carbon fixation. And then we're going to go through a bunch of oxidation and reduction reactions in phase 2 called reduction. And then over here, phase 3 is regeneration of ribulose, which means we're going to recreate RUBP. So we're going to recreate this ribulose biphosphate. So once again, we start with RUBP, we add carbon dioxide using Rubisco, and we create a 6-carbon molecule. That 6-carbon molecule splits into 2 3-carbon molecules, and then we add some energy. You can notice ATP is going into this molecule. We're phosphorylating it. And then if we look over here, NADPH is going into this molecule, and we're creating these molecules of G3P. So we create a bunch of these molecules of G3P. Now, one of these molecules of G3P comes off, and it goes to make sugar. So one of these G3Ps come off, and it goes to make sugar. And that's our 1/2 of a sugar molecule. Then this thing gets regenerated, and it takes more energy again to regenerate. If you look here, we have an input of ATP, but it gets regenerated back into RUBP. And then the cycle starts all over again. We take RUBP, we add carbon dioxide to it, and we're going to do this 3 times. We're going to add 3 carbon dioxides to 3 ribulose biphosphates, and then we're going to split that molecule, generate G3P. 1 G3P is going to come off, which is 3 carbon dioxides, or 3 carbon molecules, and then those are going to get regenerated into ribulose phosphate and then to ribulose biphosphate. So that's what happens. So we take RUBP, we attach carbon dioxide to it, we make a G3P, we pull 1 G3P out to make sugar, and then we regenerate it back. Now I want to talk a little bit about these molecules called ATP and NADPH, just as a general understanding of these molecules. So ATP stands for adenosine triphosphate, and it's the energy storage molecule for cells, and you're probably familiar with that from biology. So it's a molecule that stores energy. The big thing about this is where it stores energy. Energy is stored between the 2nd and 3rd phosphate. So this red colored bond right here, here's our 1st phosphate, our 2nd phosphate, and our 3rd phosphate. So right here, this bond right here stores all the energy. So when this phosphate is attached, this is an ATP molecule, and it has a bunch of stored energy. Now, when the bond between the 2nd and 3rd phosphate is broken, we get a release of energy. So when this phosphate's removed and this bond becomes broken, we get a big release of energy. Okay? But look at what we're left with. We're left with this here. We're not destroying this molecule. We're still left with A and D di for 2 phosphate. Right? So we have adenosine diphosphate. I'm sorry, this would be the P over here. So we have adenosine diphosphate. So 2 phosphates. We didn't destroy this molecule. We're still left with a molecule called ADP. So ADP is adenosine diphosphate. It's ATP after the energy's been used up. Here's our adenosine diphosphate. We have 2 phosphates. So now what we can do is we can take ADP, adenosine diphosphate, and we can attach a phosphate to it. So when we attach this phosphate back to ADP, we replace the energy, or we store energy. So ATP works a lot like a rechargeable battery. That's sort of the best way I can kind of understand it. This rechargeable battery is great. It's portable. We can put it in whatever we need to. It can go to one place. It can use up all of its energy there, and then it can come back and get recharged. And what happens is this phosphate's going to come off. This bond's going to get broken. Energy's going to be released. But then all we need to redo is reattach the phosphate, reattach the bond, and we can restore energy back in it. So that's sort of how ATP and ADP work. So if you have any questions about that class, that is the end of our photosynthesis unit. Feel free to let me know. I know it's a lot of information. Good luck, team. We'll see you soon. Bye.